Tumor cells frequently downregulate major histocompatibility complex class I molecules to escape detection by native cytotoxic T cells, effectively bypassing the natural surveillance mechanism of the adaptive immune system. Chimeric Antigen Receptor technology overcomes this evasion by engineering T cells to recognize specific surface antigens directly, completely independent of major histocompatibility complex presentation or peptide processing. The extracellular domain typically consists of a single-chain variable fragment derived from a monoclonal antibody, comprising the variable regions of the heavy and light chains joined by a peptide linker. This flexible peptide linker length and amino acid composition are critical for ensuring the proper folding, orientation, and thermodynamic stability of the binding domain on the T cell surface.
Specificity is determined by these variable fragments, allowing the T cell to target specific proteins like cluster of differentiation 19, which is expressed on most malignant B cell surfaces. Behind the binding domain lies the hinge, or spacer region, which provides the necessary flexibility to reach the target epitope and helps overcome steric hindrance during the cell interaction. Spacer length must be optimized based on the location of the target epitope. Longer spacers are required for membrane proximal targets, while shorter spacers are preferred for distal epitopes. The transmembrane domain anchors the receptor into the T cell plasma membrane and is often derived from cluster of differentiation 8 or cluster of differentiation 28 molecules for stability.
Intracellularly, the receptor contains signaling domains that trigger T cell activation. First-generation receptors utilized only the cluster of differentiation 3 zeta chain, containing 3 immunoreceptor tyrosine-based activation motifs. These early designs frequently failed in clinical trials because they lacked necessary co-stimulatory signals, leading to T cell anergy, metabolic exhaustion, and a lack of long-term persistence. Second-generation chimeric antigen receptors integrated a co-stimulatory domain, such as cluster of differentiation 28 or 41BB, to provide the secondary signal required for robust activation. Cluster of differentiation 28 domains provide a rapid and intense peak of glycolytic activity and proliferation, which is ideal for achieving a fast initial reduction in tumor burden. In contrast, 41BB domains promote slower but more sustained T cell persistence by enhancing mitochondrial biogenesis and memory formation, leading to longer-lasting therapeutic surveillance in patients. Third-generation receptors incorporate 2 distinct co-stimulatory domains in tandem to theoretically combine the rapid effector response of cluster of differentiation 28 with the longevity of 41BB. Fourth-generation designs, known as TRuCs, are engineered to secrete cytokines like interleukin-12 upon receptor activation, which helps to reshape the hostile and immunosuppressive tumor microenvironment. These secreted cytokines recruit and activate other innate immune cells, such as macrophages and natural killer cells, creating a broader and more comprehensive attack against the malignant tissue. The manufacturing process begins with leukapheresis, where blood is drawn from the patient and passed through a centrifuge to isolate the white blood cell fraction before returning other components. T cells are isolated from the mixture and activated using paramagnetic beads coated with antibodies against cluster of differentiation 3 and cluster of differentiation 28 to simulate natural antigen encounter. Genetic modification is achieved using viral vectors, such as lentivirus or retrovirus, which are engineered to be replication incompetent and carry the genetic instructions for the chimeric antigen receptor. These vectors integrate the receptor transgene directly into the host cell deoxyribonucleic acid, ensuring that all daughter cells produced during expansion will also express the chimeric antigen receptor. Non-viral methods like transposons or messenger ribonucleic acid transfection are emerging as safer or cheaper alternatives to viral vectors, though they vary in integration efficiency and expression duration. Modified cells are expanded ex vivo in large-scale bioreactors, where they are provided with growth factors like interleukin-2 to reach a therapeutic dose of hundreds of millions of cells. Quality control measures involve flow cytometry to verify receptor expression levels and potency assays to confirm the cells can effectively kill target cells in a controlled laboratory environment. Before infusion, the patient undergoes lymphodepleting chemotherapy to reduce the number of endogenous lymphocytes and increase the availability of homeostatic cytokines like interleukin-7 and 15. Once infused, the chimeric antigen receptor T cells migrate through the circulatory system to reach primary tumors and metastatic sites, following chemokine gradients to localize at the cancer cells. Upon encountering the target antigen, the receptors cluster at the point of contact, forming a synthetic immunological synapse that focuses the release of cytotoxic granules toward the malignant cell membrane. This clustering triggers the phosphorylation of the tyrosine motifs by members of the Src family kinases, initiating a downstream signaling cascade involving the zeta chain-associated protein kinase 70. The activated T cell releases perforin, which inserts into the target membrane to form pores, and granzymes, which enter through these pores to initiate the programmed cell death cascade. In addition to granule release, the T cell expresses surface molecules like FasL that bind to death receptors on the tumor cell, providing a secondary mechanism for inducing apoptosis. Massive in vivo T cell expansion and tumor lysis can lead to cytokine release syndrome, where systemic levels of interleukin-6 and interferon gamma rise to dangerous, life-threatening levels. Clinicians manage cytokine release syndrome using tocilizumab, a monoclonal antibody that blocks the interleukin-6 receptor, effectively dampening the systemic inflammatory response without necessarily destroying the engineered T cells. Immune effector cell-associated neurotoxicity syndrome involves the disruption of the blood-brain barrier, leading to cytokine infiltration and the activation of glial cells within the central nervous system. Treating solid tumors is significantly more difficult due to the lack of unique surface antigens, which increases the risk of off-tumor toxicity against healthy tissues expressing the same markers. The physical barrier of the tumor stroma, consisting of dense collagen and fibroblasts, prevents the engineered T cells from penetrating deep into the tumor mass to reach the malignant cells. Tumors also secrete transforming growth factor beta and express programmed death ligand 1, which bind to inhibitory receptors on the T cells to turn off their cytotoxic activity. To improve specificity, logic-gated chimeric antigen receptors are being developed, such as AND-gate systems that require the simultaneous presence of two different antigens to trigger activation. NOT-gate systems prevent activation if a specific marker found on healthy cells is detected, protecting vital organs, even if they share one antigen with the tumor being targeted. Universal or off-the-shelf cells are being developed using healthy donor T cells, which could provide immediate treatment without the multi-week delay of personalized manufacturing processes. Gene editing technologies, like clustered regularly interspaced short palindromic repeats, are used to delete the endogenous T cell receptor from donor cells to prevent graft versus host disease. Similarly, deleting major histocompatibility complex molecules from the donor cells prevents the patient's own immune system from recognizing and rejecting the therapeutic cells before they can work. Future research focuses on armoring cells with specific chemokine receptors that allow them to follow signals specifically emitted by solid tumors, overcoming the current localization challenges in oncology. Bicistronic or tandem receptors allow a single T cell to target two different antigens simultaneously, reducing the likelihood of tumor escape through the loss of a single target protein. Engineering the metabolic pathways of the T cells, such as increasing fatty acid oxidation, can help them survive and maintain function within the nutrient-poor and acidic tumor microenvironment. Safety switches using small molecule drugs allow clinicians to remotely turn off or eliminate the engineered cells if toxicities become unmanageable during the course of treatment. Modifying telomere maintenance or overexpressing specific transcription factors can prevent the cellular senescence that often limits the long-term effectiveness of adoptively transferred T cell therapies. Chimeric antigen receptors can be tuned to respond only to high densities of antigen, allowing them to distinguish between tumor cells and healthy cells expressing low levels of the marker. Synthetic Notch receptors allow the T cell to sense one antigen, which then triggers the localized expression of a second receptor or a therapeutic payload specifically within the tumor. Combining chimeric antigen receptor T cells with checkpoint inhibitors, like anti-programmed death-1 antibodies, can prevent the premature exhaustion of the cells within the suppressive tumor environment.
Emerging techniques aim to deliver genetic instructions directly into T cells already inside the patient's body, using targeted nanoparticles, potentially eliminating the need for expensive ex vivo manufacturing. Synthetic biology is rapidly refining these cellular machines, transforming the human immune system into a programmable and precision weapon capable of eradicating even the most advanced and resistant cancers.